Y'all, I'm Dr. Flatt. This is Biology 2230, General Microbiology at Dyersburg State. This is a 4-hour course with a 3-hour lab. We will be surveying the major groups of microorganisms, bacteria, fungi, algae, protozoa, and viruses, but we spend a disproportionate amount of time with bacteria. And we will be going through staining procedures essential to identifying these organisms. The simplest procedure is known as the negative stain, also known as a capsule stain. To do this, we put a single drop of nigrosin at one end of a slide, and this is a negative stain now. This is sometimes called a capsule stain. And the nigrosin is an acidic stain, and also India ink can be used for this. It's an acidic stain also. And with a capsule stain, we also counterstain by using a basic stain such as safranin. The cell membrane is negatively charged, and so when you use the nigrosin, it will pull the slime layer away from the cell wall. And then you can counterstain that with a basic stain such as safranin or methylene blue or crystal violet with that. But with this staining procedure, you can determine the shape and the arrangement, also the size. That's the first step among these morphological characteristics. It's a very simple procedure to do. It takes a little time for it to air dry, but we can actually see these cells without it completely air drying. I'm going to demonstrate this today without using the nichrome needles with this, but with those, we would always want to make sure we kill off the bacteria on the needle before we start. And then when we get through, we make sure that we kill the bacteria on that as well. These are already sterilized, and so this will work real well. And so we'll demonstrate this with using these disposable loops with this. And the first thing we do now is put a single drop of nigrosin again at the end of the slide. This is the only staining procedure in which we will do that, by putting it at the end of the slide, like this right here. Okay? Then we lift up the edge of the plate, treat everything as if it's potentially pathogenic on this. We lift this up, and then we put that right back down. Let me choose this one right here. Others are kind of closed up a little bit. And then spread this out in this drop of nigrosin like that. Then we throw this away. I have a trash can here. We'll always be careful with these stains. These are highly corrosive. We take a second slide now, we put it in front of that, and we push that forward and bring it back, and we let that air dry. And with that, we can determine the size, the shape, and the arrangement. Normally, we would be using the Bunsen burners on this to kill the bacteria, since we can use those over and over again, but this is very convenient here. Let me point out again that this is a negative stain in which we're staining the background instead of the cells. We follow this up with other staining procedures. The most important stain we do is the Gram stain, and that's also a differential staining technique. Sometimes we refer to the negative stain as a capsule stain. When the slime layer is sufficiently thick, it's referred to as a capsule. But in order to actually see the capsules, to see the halo with that, we use the nigrosin or we can use Congo red dye, something that will not penetrate the cell. This is something that you can do with an inexpensive microscope and quickly determine the shape and the arrangement of the bacteria. India ink works real well with this. We use nigrosin, but they're acidic stains. With the other stains, we use a basic stain with that. So that with a capsule stain, we'd carry this a little bit further, but it's still a very simple procedure. So when this is sufficiently air dry, then we would examine that with a compound microscope. We would search first with the low power, bring it into focus. If it's very small, like for Escherichia coli, then we would use oil to bring that into focus and magnify that 1,000 diameters. So You have the exercise in front of you there that we'll be doing. The next thing you would do, you would scrub the tabletops as I've done up here. Then you would get the microscopes. These are compound microscopes. They're top of the line, and so you can go up to 1,000 diameters with these. Get well organized before you start. Make sure you have all the materials that you need, and then do the negative stain. Then this will be coming up again. Later we'll be doing unknowns, and this will be the first step among many steps. This only determines the morphological characteristics. We would follow this up with biochemical characteristics.